So let's go ahead and take a look at active transport now. In active transport, this is going to require energy. Okay, active requires energy. Remember that energy is going to be in the form of ATP or adenosine triphosphate. Okay, active transport may use transport proteins if you're talking about molecular pumps. The example of a molecular pump that we need to be familiar with is called the sodium-potassium pump, and we will take a look at that in just a few slides. Okay, so now in active transport, it does require energy, and it may use a transport protein, and the reason it requires energy is because it's going to move materials from an area of low concentration to an area of high concentration, and we call that moving against the gradient. So facilitated transport, osmosis, diffusion, those will go with the gradient, so they're going to go high to low, no energy required. But in active transport, we are going against the gradient, and that is going to be from low concentration to high concentration, and that is why we need to use energy. In some cases, we're going to use molecular pumps, and in other cases, we're going to use the cell membrane to actually bring the material in, and we will talk about that in a moment. So the first one we want to look at is the molecular pump, and I mentioned to you that we are going to know about the sodium-potassium pump. So if you take a look here, we've got sodium ions, this Na+, all right, and we have our ATP, adenosine 1,2,3 triphosphate. So the ATP is going to bind a phosphate. This is called phosphorylation. It's going to bind to our protein. When it does, it's going to accept these sodium ions. It's going to accept 3 sodium ions, and it's going to force them from an area of where they are in a lower concentration to an area of higher concentration. After that reaction takes place, 2 potassium ions will bind to that protein as well. All right, and those will be pumped into the cell, again, from an area of low concentration to an area of high concentration. Okay, this is going to have something to do with a process called action potential. All right, so now let's take a look at an animation of this very same process. So here we have our biological membrane, our phospholipid bilayer. We've got our embedded protein here, our integral protein. And this is going to be used to move the molecules from one side of the membrane from an area of low concentration for sodium. So the sodium is going to be pumped out of the cell. And in return, 2 potassium ions are going to be pumped back into the cell from an area of low concentration to an area of high concentration. And to do this, we're going to need some energy in the form of ATP. So what's going to happen is the ATP is going to bind to the protein. It's going to add a phosphate group there. Again, that's called phosphorylation. Okay. The ATP then becomes something called ADP, which is adenosine diphosphate. Remember, we had the phosphate onto the protein. That's going to help us to get the protein to open up, the protein channel. And then sodium ions are going to be pumped out, and potassium ions are going to be pumped into the cell against their gradients, and then the protein channel closes. Okay, that protein channel can be used again, but again, we would need another ATP molecule to make this reaction occur. This was called the sodium-potassium pump. So now we're going to look at the next type of active transport called cell membrane mediated transport. And we have 2 types of cell membrane mediated transport. The first type is called exocytosis, and the second type is called endocytosis. Okay, and both of these are dealing with the movement of large molecules by the action of membrane movement. Okay, so the membrane is going to surround the material. Okay, and in endocytosis, bring it in, and exocytosis, that membrane is going to bind to the outer membrane of the cell and expel waste or chemicals. So let's take a look at the 3 types of endocytosis. The first type is called phagocytosis, and this is when we are bringing in large particles like food. Phagocytosis literally means a process, osis, of the cell, cyto, feeding, phago. So it means feeding cell process, or process of the cell feeding. And if you notice here, this is the phospholipid bilayer. It is surrounding the particle, and then that particle is broken off and forms a vacuole. So this right here would be a food vacuole. Notice that the material that makes up the membrane around the vacuole is the same material that makes up the plasma membrane, this phospholipid layer. The next type of endocytosis is called pinocytosis, and this one is a process of cell drinking. Okay, so these are water and dissolved materials. Same process, the cell membrane surrounds the material and it breaks off and forms a vesicle or a vacuole. Remember, this is also going to be made up of the phospholipid bilayer. The third type of endocytosis is called receptor-mediated endocytosis. Receptor meaning there are things on the membrane, receptors, that will fit certain molecules. Okay? When they bind to those molecules, a series of reactions occurs, and the membrane surrounds the material and brings it into the cell. Okay, again, these are different types of endocytosis, which is a process of cell entering, bringing something into the cell. So let's go ahead and take a look at a short animation. So here we have our cell in yellow. We have our, well, this is my favorite food anyway, favorite food, pizza. Okay? So what's going to happen is the cell moves towards the material. Okay? You get an indentation in the membrane. It starts to surround that material and then ultimately incorporates it into the cell. So this would be bringing food material in. This would be phagocytosis. The next type of endocytosis was pinocytosis. So if we take a look here at pinocytosis, the difference is simply the material that's being brought in. Again, here's my second favorite food, or I guess my favorite drink, and this would be coffee. But for the cell, it would be water or some type of dissolved material. Now let's go ahead and take a look at exocytosis. Exocytosis means a process of cell exiting. Okay, so this cell exiting could be getting rid of waste materials. Okay, but it could also be the transportation of secretions and hormones that were produced inside of the Golgi body. All right, could be those being secreted to the outside of the cell, and then those secretions can move wherever they need to go to do their job. It could be hormones and things like that. All right, so this right here is going to break off into a vesicle. In this case, it is a secretory vesicle. Okay, these could be different types of proteins. Again, these could be hormones. And as they move towards the membrane, they bind to the membrane. Notice that the materials are the same. They bind to the membrane and ultimately release their contents to the outside. Over here, we have something called a transport vesicle, and this is where we've made other types of materials. This could also be, again, waste materials moving towards the membrane. Once it binds to the membrane, it's going to release its material to the external environment. And again, we have a short little animation here. So we have our yellow cell. Inside that yellow cell, we have a vesicle, which is full of, you could say, hormones or different secretions, or this could be waste material. And what's going to happen is that vesicle will move towards the outside of the cell. That vesicle will fuse with the cell membrane, and it will release its contents into the environment. Okay, again, as it does this, it will reform the membrane, and we have now seen what exocytosis looks like. So, as a quick review for cell transport, we have passive transport and we have active transport. For passive transport, we have diffusion, we have osmosis, which is a specialized type of diffusion where we have water moving from an area of high concentration to an area of low concentration. And then we also have facilitated diffusion, and the difference between facilitated diffusion and regular diffusion is just the addition of a molecule, a channel protein, to help move materials across the membrane. Okay, in all of those, we are still talking about going from high concentration to low concentration without the use of energy. Then we also have active transport, and for active transport, we have molecular pumps, and we also have cell-mediated transport. In all cases for active transport, you are using energy. You're moving materials from an area of low concentration to high concentration when you're dealing with molecular pumps, and when you're dealing with cell-mediated movement, you're dealing with the movement of materials into or out of the cell via movements of the cell membrane. And in these cases, again, you are going to use energy. You are moving from low to high concentration. You may need carrier proteins in the case of molecular pumps, or not if you're talking about cell membrane mediated transport. Okay, the last slide we want to show is simply talking about the diversity of cellular life. And when we're talking about the diversity of cellular life, I just want to remind you, we have talked about these organisms. Okay? Remember, we have organisms that are unicellular and others that are multicellular. And remember that unicellular organisms have to carry out all of the life functions on their own. So all of these cell transport models that we just discussed, these unicellular organisms have to be able to do. If you're talking about multicellular organisms, remember that they are going to contain specialized cells which have to work together, but they have cells that perform specific functions, okay, and do not have to do everything on their own. You have certain cells that are involved in reproduction. You have other cells that are involved in movement. Other cells may be involved in transport of materials. Okay, so hopefully this helped you guys out, and if you have any questions, make sure you bring them to lecture so that we can go ahead and go through them, and I will see you guys in lecture.